Okay. First of all, thank you to the organizing committee. It's my first conference and I've had a lot of fun, so thank you. And then I would like to talk to you about image-based yield prediction for dull fescue. So as we've heard this morning, high-throughput phenotyping can have some advantages over traditional phenotyping. For example, it can decrease the amount of labor and time needed to phenotype a field. And in the case of visual traditional phenotyping, it can also be a little bit less objective. So a high-throughput phenotyping approach can be more objective, which can increase the reproducibility and comparability of assessments. But what I wondered is whether these high-throughput phenotyping assessments could also be more accurate for certain traits. To test this hypothesis, I used the dull fescue breeding program at Ghent University. I used, more specifically, a progeny assessment trial of more than 4,200 dull fescue individuals. For the ones who don't really know dull fescue, it's a very interesting forage crop. It is more robust to the effects of climate change in northwestern Europe than its more popular competitor, perennial ryegrass. And it also has a very high yield potential. Yield is also the trait I was interested in, more specifically dry matter yields. And I evaluated it in this field in 3 ways. First of all, an experienced plant breeder evaluated each individual with a score from 0 to 5 based on its yield. Secondly, I took RGB images using a drone of the field. And thirdly, I took destructive yield measurements as a ground truth variable. To analyze this data, I created 3 models. First of all, to evaluate the assessments of the breeder, I created a simple linear model with a breeder score as a classification factor with 6 levels. Then I evaluated my high-throughput phenotyping approach, so the RGB images, with 2 different machine learning algorithms, a random forest and a convolutional neural network. I wanted to try both of these because I wanted to try one approach with manual feature extraction and one with automated feature extraction. Manual feature extraction allows for more control over the prediction process, but also means you have to have some domain knowledge to pick the best features. So I was very curious to compare my own chosen features and the automated features of the CNN. I will immediately give a little more detail about my chosen feature extraction algorithms. I first started with some general image features that can be used in all type of computer vision problems that base itself on colors, shapes, and textures in the image. Secondly, I read some papers using vegetation indices, which I also thought was a very interesting idea, so I chose 4 RGB-based vegetation indices. And lastly, I realized that it would be logical if the number of green pixels in an image would also correlate with the yield of the plant, so I also made 2 green pixel counting algorithms as extra traits. As I've mentioned before, for the CNN, I did not have to do any feature extraction, but I did need to choose an architecture. As we've heard earlier, the architecture is kind of the structure of the model, so the number of layers, the type of layers, the exact specifications of each layer, and so on. As you can imagine, this is computationally intensive to optimize, so I decided not to do that myself. There are various optimized architectures available online, which work very well. So I tested 4 of these based on their comparability with previous studies, their number of parameters, and their accuracy on ImageNet, one of the most important benchmark data sets for computer vision. With these pre-trained architectures also came a set of newly trained weights and biases. These weights and biases are, of course, trained on the ImageNet dataset for these pre-trained architectures. But because many features in computer vision are fairly general, such as the color and the shape and so on, you can often use the parameters of one dataset for your own dataset as well. So I was curious whether that would be possible for my dataset, so I also tested the pre-trained parameters and compared them with completely newly training the entire architecture. Well, the parameters in the architecture, not the architecture itself. So now I had 3 models: a simple linear model to evaluate the breeder score, and a CNN and random forest to analyze the RGB images and see how well they could predict dry matter yield. However, while doing this, I realized that knowing the exact dry matter yield is not always necessary. In a breeding context, it can be sufficient to simply know which plants perform best for selection. So that is why I also created a binary classification model, well, 2, a CNN and a random forest, to identify the top 10% highest yielding individuals in my dataset. On to the results. Firstly, for the random forest, I was curious about my features and whether they did well. So I checked out the feature importance, and over here you can see the top 5 most important features for both the regression and the binary classification problems. I've highlighted the 2 green pixel counters because they were important in both models. And you can also see which pixels they highlighted in the images. 2 other features that were important in these top 5s were color histogram number 68 and number 4, which try to capture the ground around the plant, as you can also see in these images. All of this to say that the random forest based its predictions on logical, biologically meaningful features, which to me was very reassuring. Definitely for practical applications, it's nice to know that the predictions kind of make sense. And then for the CNN, I compared the architectures and the pre-trained parameters with newly trained parameters. As you can see, the pre-trained parameters in the dark green bars perform worse than the newly trained weights and biases in the light green bars. I think this might be because the pre-trained parameters were trained on the ImageNet dataset, and that dataset is very large and of a very high quality, but it is not targeted to my problem specifically. It doesn't contain 4,200 images of tall fescue for some reason. So therefore, I think my smaller dataset that was of a lower quality still performed better than using the pre-trained weights and biases. And then to compare the different architectures, when we look at the light green bars, so the newly trained parameters, there's really not that much of a difference. DenseNet 161 was slightly better and consistently better, so we continued with that one, but it seemed like all of them were quite suitable to this dataset. To finish, the main results for dry matter yields, for the dry matter yield predictions, you can see in the green bars that the machine learning approaches performed better than the model using the breeder score. So in this case, the machine learning models were better, so the high throughput phenotyping approach that I chose. I think that might have something to do with the objectivity I talked about before. I think the breeder score might be influenced by non-yield related factors, some of them useful, for example, signs of disease, but other may be a little bit less useful, such as subconscious biases like the performance of the previous plant or anything like that. And then for the binary classifications, both models were very good at identifying the best performing crops. So I thought that was very interesting for practical applications. And lastly, the CNN and random forest performed fairly similarly. CNN did outperform the RF, which makes sense because it is known to be the state of the art for many computer vision problems. So that was a logical result. To conclude, using pre-trained parameters in my case was less good. The pre-trained parameters were less suitable than my newly trained parameters. The CNN and random forest performed similarly, but the CNN did defend its reputation as state-of-the-art. And the CNN and RF outperformed the breeder score in predicting dry matter yields. So in my specific case, the high-throughput phenotyping approach was more accurate than the breeder's eye. But of course, to really answer this question, I would have to test more environments, more genotypes, more crops, everything more, really. So the quest continues. To finish, if you would like some more information, you can check out my paper. You can contact me or my research group on LinkedIn, or you can, of course, ask a question right now.